Hi, in this PPT we'll be seeing about regulation of gene expression. The first slide shows about the structure of immunoglobulin and we'll be seeing about the expression in heavy chain. First thing, multiple germline gene segments. In human germline DNA 51, VH27D, 6JH40, VK, 5JK, and these are present. In mouse, these some other gene segments are also present. They contribute the antigen binding sites and antibodies. The combinatorial of VDJ joining. In humans, the ability of the gene 51 VH segments to combine with any of the 27 DH segments and any one of the 6 segments allows a considerable amount of heavy chain gene diversity to be generated. So it is equivalent to 120 possibility combinations. Total antibody combining diversity in human to well over 10 part 10. The third one is junctional flexibility. It increases the enormous diversity generated by means of V, D, and J combinations. They can lead to many non-productive rearrangements, but it also generates several productive combinations that encode alternative amino acids to each coding joints, thereby increasing the antibody diversity. And this picture clearly depicts the lambda chain of DNA and kappa chain of DNA. First, lambda chain multigene family. Functional lambda variable region contains two coding segments which are separated by non-coding DNA sequence in unarranged germline DNA. J for joining and J lambda 4 is a pseudogene for defective gene that is incapable of coding proteins denoted by psi. Lambda chain has three subtypes, lambda 1, lambda 2 and lambda 3. They also contain many pseudogenes. Then kappa chain multigene family. This multi-gene family contains approximately 85 VK and 5 JK, one pseudogene, and one CK gene segment. In humans, approximately 40 kappa chain V and 5 JK and 1 CK gene segments are present. And this picture clearly shows about the arrangement of heavy chain and which is later transcripted and mRNA is formed and by splicing and other processes are also formed and later the gene expression in heavy chain is expressed. The heavy chain family contains V, D, J and C segments. The arranged V, D, J gene segments encode the variable region of the heavy chain. Each gene segment is preceded at its 5' end or the linear peptide that guides heavy or light chain through endoplasmic reticulum. Signal peptide is cleaved from the nascent light and heavy chains before assembly of the final immunoglobulin molecule. PDJ rearrangements in heavy chain DNA. This process requires two separate rearrangement events within the variable region. DH segment joins first to a JH segment. The resulting DH-JH segments joins a VH segment to generate a VH-DH-JH unit that encodes the entire variable region. Rearrangement of produces a short L exon, an intron, and join VJ segment, another intron, and a series of C gene segments. And this is all about expression of genes in heavy chain. Thank you.